Good afternoon, everyone. My name is Molly Howell, and I am the Immunization Program Manager for the North Dakota Department of Health. And today I am here to introduce Dr. Paul Carson, who will be presenting this webinar on COVID-19 vaccine and what you need to know, or at least what we know so far. Dr. Carson is an infectious disease physician with over 25 years of clinical experience. He is also a professor of practice at the North Dakota State University Department of Public Health and medical director for the Center for Immunization Research and Education, also known as the CIRE. Just so you're aware, the session is being recorded and will be available for continuing education credit. For information regarding continuing education credits, you can contact NDSU at ndsu.cire@ndsu.edu, and hopefully someone will put that in the chat for everyone in case you have questions. And there will be time for questions at the end of the webinar, so please type your questions during the presentation into the chat box. And you can also email them in anonymously to that email address. And otherwise, I am going to turn it over to Dr. Carson.
Thank you, Molly. And good afternoon, everyone. Pleasure to be with you, and looking forward to going over what we know so far and what we're learning about vaccine for SARS-CoV-2, a very hot topic, lots of questions and lots of issues around it. I'm glad to have Molly with me, as well as a couple of our program managers from our center, Kylie Hall and Lauren Dibson, and we all spend a lot of time on this, and I may be tapping them as well for some of the questions that might arise. So I'm going to share my screen and we'll get started. Molly or Lauren, do you guys want to call for the poll questions first?
Yes, I can happily do that. If you want to go to the next slide, I think we have a slide just for the polling, and then Kylie can put forth the questions. Mm-hmm. So we would like to poll all of our participants for some information. If you could answer this question, and that is, what is your professional designation?
Looks like a lot of nurses and pharmacists and a handful of physicians and others, some students, so that's great. How about the next? Looks like we're ready for the next one.
So our next question is, if the FDA authorized a vaccine to prevent COVID-19 today at no cost under emergency use authorization, also known as EUA, would you agree to be vaccinated?
Okay. And how about the next one, Lauren?
Next, what would be the absolute lowest possible rate of a severe adverse event, also referred to as an SAE? This would be considered an event following a vaccination, which would be life-threatening or required hospitalization, in a COVID-19 vaccine that you would find acceptable.
And Lauren and Dr. Carson and Kylie, I just wanted to mention people are trying to join, but they're being told it is full. It
Suppose we have
must be,
capacity for 1,000, so
Yeah.
it shouldn't be.
we're getting a lot of emails and concerns, so I don't know.
Do we have Wendy on that can maybe try and troubleshoot that?
I will contact her right away.
While we're doing that, can we maybe do the next poll? I think we only got maybe one left here.
So what do you think is the relative risk of acute respiratory distress syndrome in a laboratory confirmed COVID-19 case compared to a patient with influenza?
And I can't remember, Lauren, is that the last one? Is there one more after that?
I think there is one more following this one.
Okay. Last question.
Lastly, yep. If someone becomes sick from COVID-19, what do you think is their overall chance of being hospitalized? I contacted Wendy, so we'll see what she has to say.
Okay, so that's helpful. That's interesting. Hopefully you'll maybe learn a little bit from this presentation that might help inform some of those answers a little better for you. Why don't we go ahead and end the polling? And I don't know if you need to share those or if you have those stored for later. And if I can proceed on, is that okay, Lauren?
You are ready to go.
Okay, great. All right, thank you. So does everybody see my screen okay? Can you give me the thumbs up, Molly? You see that? Okay, great. So the objectives today are a handful here. We're gonna just very briefly go over some of the virology and a bit more about the epidemiology of the SARS-CoV-2 virus and where we're at right now. Spend a decent amount of time on describing the development, licensure, approval process, and possible distribution of a COVID-19 vaccine. Want to go at the end because you all, the majority of you on this webinar are healthcare professionals. And when you poll the general public, it is still healthcare professionals that tend to be the most trusted people for getting advice, or when people want to get advice on matters of healthcare, including things like vaccination. So we hope to be able to help you know how to confidently address patient concerns with the COVID-19 vaccine. And then we're going to describe North Dakota's role in planning for the pilot distribution of the vaccine. So just briefly, a little bit about the virus. Most of you have heard a lot about this, but we know that coronaviruses are from a large family of viruses, and some of these cause illnesses in people, and others only infect animals. We have 4 common coronaviruses that have been circulating in humans for a long time, probably centuries. These are, by their letter and number names: NL63, 229E, OC43, and HKU1. These typically cause the common cold, and most of us have been infected with one or more of these viruses in our lifetime, usually in childhood. And we know that some coronaviruses will infect animals and then spread to people. Bats tend to be a very good reservoir for a number of viruses. They may jump to other mammal species, which then makes it easier for them to evolve to make a jump into humans. That is believed to have happened with the Middle East Respiratory Syndrome, or MERS, the original SARS, now called SARS-CoV-1, standing for Severe Acute Respiratory Syndrome virus, and now most recently SARS-CoV-2, the cause of the disease COVID-19. So these three were very different than the common human coronaviruses, much more lethal. The MERS having a case fatality rate, so if you become sick with MERS, up to around 30%, and with the original SARS-CoV-1, around 12 to 14%. That led a lot of attention to these viruses, and people recognizing, scientists, public health officials, researchers recognizing these actually pose an existential threat to us. If you had one of those with that high of a lethality that was very transmissible, that this poses major pandemic potential. And so they've been a hot focus of research for the last decade and a half. And when we look at vaccination as a means to try and prevent this, we can see that we're embroiled in a lot of controversy, information and misinformation. Here's a person not only afraid of the vaccine, afraid of getting microchipped with the vaccine, and we see some of these memes kind of running around on the internet. But more importantly, not too dissimilar from the poll that we just took of all of you, the Pew Charitable Trust has a research center, and they polled the general public back in May and found at that time people were about 72% inclined to receive a vaccine if it were to become available. Repolling towards the end of September found a substantial drop in the percentage. Now almost 49% saying they either probably will not or definitely would not get the vaccine. So how do we kind of think about this, and how do we kind of do this math? So whenever we consider a vaccine, both for licensure and for our own individual choices, we are trying to weigh a set of risks and benefits. The risks that might come with any drug or treatment or vaccine, because nothing that we take or give to try and prevent disease or cure disease or manage disease comes with zero risk. So we're trying to gain whatever benefit we can with the least amount of risk, but we're counterbalancing that against the risks of the virus and the pandemic and its spread. Well, when you look at how we as humans approach that sort of calculus, we're not very good at it. If you're interested in this sort of thing, I highly encourage you to look at a couple different books out there if you like to read about this sort of stuff like I do. One is called Thinking Fast, Thinking Slow by Daniel Kahneman. He's a research psychologist at Princeton who built a career on sort of how we approach things with our mind. Another book is called Innumeracy, which is about how well the majority of us are sort of manipulating kind of basic math to assess risks and threats to us. And the bottom line is that we tend to use a lot of what are called heuristics. These are sort of rules of thumb, kind of quick off the ground thinking that tend to work well in sort of everyday life, but tend to be a lot less accurate and not very good for when matters are more complex. And we're prone to a lot of biases, and this includes those of us in the healthcare profession. We are not immune to this at all. We tend to have a bias that's sort of sometimes called a do no harm bias, meaning I'm more comfortable with the idea of a bad thing happening to me from just nature or fate than a bad thing happening to me because it was a decision that I made to do it. But that's actually not very good thinking, because it's a bad thing either way. So bad things can come from inaction and bad things can come from action. And you really want to do the calculus for both and not be swayed inappropriately by a bias towards, I'd rather just take my chances with fate. We're also more comfortable with known risks than ambiguity. So, and we can see how this plays out in the vaccine here in a minute. We're also prone to something called availability bias. We conflate things that we remember easier. If I hear a story about a kid who got a vaccine and later developed autism, that can weigh heavier in my mind, and it actually we tend to overestimate the risk based on anecdotes that we've recently heard. And lastly is a bias called compression bias. We tend to overestimate rare risks and underestimate common risks, especially as they become more familiar. We see this frequently with influenza. We're very, by and large as a population, kind of blasé about getting the flu vaccine. That's a much greater risk to us than almost any of the other things we tend to worry about, flying in an airplane, getting bit by a snake, whatever. Influenza poses a much greater risk, and it's so familiar to us that we don't do anything. So we have to be aware of these rule of thumb types of thinking and biases when we try and do this calculus of risks and benefits. So let's take the first set of risks here, which is what are the real risks to us from the virus? Most of you know now we are in sort of a third wave of the pandemic. We are now over 9 million cases in the United States. That's a little over 2%, almost 3% of the population has been infected with this, and that's the known diagnosed cases. It's probably much, much greater than that. And we have over 230,000 deaths from this in the United States. And if we look at excess mortality, so some people have mistakenly sort of thought, well, this isn't much different than influenza, or maybe we're overcounting these, or maybe we're conflating those numbers. If you just take a look at all-cause mortality, people dying from any reason in 2020, which is the red line, compared to the prior 5 years, 2015 to 2019, this is data that the CDC has tracked for decades, we can see that there's a lot more deaths going on in almost every age group, even down into that 25 to 44-year-old age group. A lot more deaths in 2020 than in the prior 5 years. Now, you can say maybe that's because of things like lockdowns at the beginning and people not going in to get care for their heart attack or so on. And that is part of it. But actually, when you look at this and attribute also what we know about COVID diagnoses, the vast majority of this, over 90% of these excess deaths is directly attributable to COVID, and probably even another chunk of those that we haven't measured because testing wasn't as available at the beginning and people may not be getting diagnosed. And if we hone down in on North Dakota, the picture is not very pretty, as most of you know lately. So the top graphs here look at the worst states for cases per capita. North Dakota was leading this list up until I think today or yesterday, and South Dakota just bumped us out into second place. But we have been neck and neck with South Dakota for the worst number of cases per capita. Now you can say you can blame that in part on our aggressive testing in North Dakota. South Dakota has very unaggressive testing, and we are kind of on par with each other. And we're in other company here in kind of the West and upper Midwest with Wisconsin, Montana, Wyoming, and Idaho rounding out the list. And if we look at the places with the highest daily reported deaths per capita in the last 7 days, North Dakota has led this list, I think we're probably entering the third week in a row for leading the number of deaths per capita over the prior 7 days. So not records that we want to hold. And if we look at hospitalizations per capita, I didn't put that on here. We're in number 2 there as well. We're in second place with South Dakota being just ahead of us for hospitalizations per capita. So it's not just counting deaths or counting cases because we test so well. We have objective measures of very high numbers of hospitalizations and very high numbers of deaths. Well, if we compare COVID-19 with similar influenza epidemics of the past, if we look kind of on a relative scale of clinical severity and a relative scale of transmissibility, this is kind of where seasonal influenzas range in sort of relatively low severity, relatively low transmissibility. They have a reproductive number, if you've kind of heard that term, the R naught, how many new cases you get for a single case, somewhere between 1 to 1.5 typically. The pandemics of 1957, 1968, and the more recent 2009 pandemic had more transmissibility, and these ones more severity. And you compare those with the sort of the granddaddy of them all in the last century, which was the Spanish flu, the 1918 pandemic, which had higher transmissibility and very high severity, lots of deaths associated with that. Where does SARS-CoV-2 or the COVID-19 rank here? It's a fair amount more transmissible or contagious than the 1918 Spanish flu, and it's getting in the ballpark of the same severity when we start looking at things like case fatality rates. This is the real deal. We have to get well past the nonsense of this is anything similar to our common flu. And if you math this out with the worst case scenario, which would be we keep marching along till pretty much everyone in the population who is susceptible gets infected, and we don't know where that number tops out. There's theories that it tops out between 50 to 70% with an alleged herd immunity, which we actually don't know if we can accomplish that. It may top out closer to 90%. We saw that in the Marion County Prison in Ohio when it swept through there in about 3 weeks and infected 90% of all the prisoners there. So if we have 328 million people in the US with somewhere between 50 to 90% of the population ultimately susceptible and ultimately infected over, say, the next year, that maths out to about 164 to 295 million infections, with an infection fatality rate that's been hovering pretty steadily around 0.6 to 0.7%. That equals a total mortality in our country of 984,000 to 1.8 million. Numbers that would exceed the Spanish flu at 650,000. Of course, we have a lot more population now than we did in 1918, but this would be one of the greatest pandemics on record. Now, some people will argue, well, we just got to muscle our way through this. Aren't we just kicking the can down the road? I mean, don't we have to just sort of face up and just put our shoulder to the wind and deal with this? Well, actually, I would say kicking the can down the road is a very good strategy, because we hope not far down the road are vaccines and better treatments. And there's a plethora of treatments in the pipeline working their way through phase 3 studies, and many, many vaccines, which we're going to address now. So my plea would be, yes, we need to keep doing mitigation as best we can to try and get to these better treatments and vaccines so that we have a much better chance of doing better with this. So kind of trying to put this again in a bit more perspective on where things rest with risk and with things like a vaccine, looking at how often someone might develop shingles, for example, in their lifetime, and of course we can get a vaccine for shingles, it's about 1 out of 3. Dying from COVID, if you do the math that I just showed you there, ultimately that might be 1 out of 300 people dying of COVID if this just keeps marching through with no better treatments and no vaccines, and either fast transmission because we do no mitigation, or stretching it out over more time by masking and distancing and so on. Dying from choking on food, about 1 out of 2,600. Dying of the flu, about 1 out of 4,000 in your lifetime. Dying of a lightning strike, about 1 out of 161,000. What's the risk of, like, for example, a bad outcome from a vaccine? One example would be Guillain-Barré syndrome or anaphylaxis from vaccination. That's about 1 to 2 out of a million. But we fear that sort of big, worrisome problem, and it's way out of balance by many other things that we're much more likely to face. Putting this in a little more perspective, this is the risk from a number of different surveillance studies around the world of either being hospitalized or dying from COVID. Infection, not becoming necessarily sick, just infected. So this would be taking all comers of asymptomatic, mildly symptomatic, severely symptomatic people. And if you take 100,000 people infected with COVID, this is the breakdown of how many will land in the hospital. So even for a 20 or 30-year-old, you're talking about 1,000 to 3,000 plus people out of 100,000 infections will land in the hospital. And when we talk about death, we're talking 30, 80, 160, 600, up to about 7,800 deaths per 100,000 infections, all comers infected here. if they get a COVID-19, if they get a SARS-CoV-2 infection. Contrast that with some of the more serious adverse events that we see with known vaccines. Again, anaphylaxis from all vaccines is about 0.1 per 100,000. Now, look at this table and look at how many hospitalizations and deaths per 100,000 when you think about these risks. One of the more common problems that we can see is intussusception in infants with the rotavirus vaccine. That's about 1 out of 100,000. Even with the really bad rotavirus vaccine that we had previous to this better version, that was about maybe 5 out of 100,000. Compare that again with what you see with a COVID-19 diagnosis. Measles, mumps, rubella, idiopathic thrombocytopenic purpura, one of the more worrisome side effects from a measles vaccine. That's about 2.5 per 100,000. Most people do fine with that and it resolves, but it can lead to some bleeding complications rarely. One of the scariest vaccines we give, yellow fever vaccine, which is a live attenuated yellow fever virus, which can cause some really severe complications. One thing called viscerotropic vaccine viral syndrome and an encephalitis. These range around 0.4 per 100,000. And we worry a lot about these. Again, compare that with what you might see with SARS-CoV-2. Smallpox, encephalitis, 1 out of 100,000. So these are some of the worst and most common severe adverse events with known vaccines. Well, what kind of study numbers are we looking at to try and assess the risk from some of the COVID-19 vaccine candidates? I put your attention at the bottom here to the Moderna vaccine, the Johnson & Johnson vaccine, the Pfizer vaccine, which are making their way through late stages of trials right now. Each of these have a 2-dose series. In the phase 3 here, the lowest of these, Moderna, which has just completed its enrollment, got a little over 30,000 subjects enrolled. Johnson & Johnson is aiming for 60,000. Pfizer is aiming for 43,000 in there, and they're getting close. They've got about, I think, 37,000 enrolled now. That's actually more than most of the prior trials we've done for things like inactivated polio or the diphtheria, tetanus, pertussis, or Gardasil, or Prevnar, where you see, anywhere as low as 2,000 to a high on the Prevnar of 49,000 in these trials. So these are robust phase 3 high enrollment goal trials. What will those sorts of numbers detect? So if you look at something that's like a bad thing happening in the background population, let's say the example of transverse myelitis, which you heard about, we'll talk about in a second with one of the vaccines. That's about a 1 out of a million in the background population. If we want to find something 5-fold higher than that, well, we're not going to get that kind of enrollment, right? That's going to be hard to detect. We might pick up it, if that's a problem, 100-fold higher. That's why we do ongoing surveillance after these vaccines are released. So very hard to pick up these 1 in a million events unless they're way higher. But we can pick up 5, 10, 100-fold higher things in 1 out of 10,000 or 1 out of 100,000 events. We're well in the range here of those trials picking up those sorts of things, even at levels as low as 5-fold higher, even maybe 2-fold higher. We can pick up 1 out of 10,000 type of numbers fairly readily with the size of trials that are being done. Well, you may hear from people, I hear this not infrequently with people who have concerns, well, I need to see the long-term results. 2 months, 6 months, 1 year. I mean, what about what happens, 2, 3, 4, 5 years down the road? Well, okay, I'd like to see that too. That makes things very unrealistic for getting any kind of a vaccine, distributed if you have to wait that long to see all the long-term effects. But what this young lady needs to think about as well is what are the long-term effects of the virus and not doing something? And here again, that sort of thinking is displaying ambiguity, more comfortable with known rather than unknown risks. That idea of doing no harm, I'd rather take my chances with not doing something than take my chances from doing something. and this kind of idea of compression, overestimating rare risks and underestimating common risks. So for a bad thing to happen way out is going to be an extremely rare event. And you need to balance this against what's the long-term outcome from COVID, right? So we're now here reading more and more and more about COVID long haulers. This is one study that was just published in the journal Infection on post-discharge symptoms at 4 months. 55% still experiencing fatigue, 42% still experiencing shortness of breath, 34% still experiencing complaining of things like memory loss. When you're doing that math on that rare possible long-term side effect, you at least need to balance that against what's the already known long-term effects that we're starting to see from COVID. And it's not just this. Think about some of the other data that's coming out on this. We're now seeing evidence of myocarditis, heart inflammation, and myocardial injury, even in healthy young athletes who have recovered from asymptomatic or mildly symptomatic COVID. Well, what does that mean for the long term? Are you willing to take chances with inaction on that? This was a study from Ohio State where they looked at 26 athletes from 5 different sports in this D1 school who had come out of their COVID-19 quarantine 11 to 53 days after recovery, and they did cardiac MRI scans on them. 73% had been asymptomatic, 27% only had mild symptoms. 15% of these met criteria for myocarditis, and an additional 31% had what's called late gadolinium enhancement on T2. That's a sign of prior myocardial injury. What does this mean for the long haul? When you balance those risks of long-term side effects of the vaccine, it must be balanced against what are the risks from even asymptomatic or mildly symptomatic in an otherwise healthy appearing athlete. We don't know yet. We're only just learning. Moving on from that, just a word about herd immunity. So vaccines work in 2 ways. They work at the individual level. They protect you as an individual, but as or maybe even more important is building up a population level immunity to where vulnerable members who either can't be vaccinated or don't respond to the vaccine will be protected by the rest of us. That threshold where we hit that, where you have enough of the population immune, is dependent on the contagiousness of that pathogen, the R naught. And it's a simple formula, 1 minus 1 over R naught. We think the R naught is somewhere between 2 to 3 for SARS-CoV-2. That puts us at a 50 to 60% herd immunity threshold, meaning we think 50 to 67% of the population immune to reach this threshold where the virus can't really spread very well. It does not get a foothold in a population. Contrast it with like measles, which has a R naught of like 14 or 15. We need 95% of the population immune before we will see herd immunity threshold. Now, that means they're immune. And when we're looking at vaccines, that's a combination of the percent who are vaccinated times the percent vaccine efficacy. So if the vaccine is 100% efficacious, we may only need 50% of the population vaccinated. Very few vaccines make that high of a bar. If we have a vaccine that's only 50% efficacious, we need everybody vaccinated. So it's probably going to fall somewhere between that. Where has the FDA set the bar for efficacy with this vaccine? They've set it at 50%. They're not really going to look at approving a vaccine unless it meets at least that 50% efficacy threshold. And that's efficacy at preventing infection, at least symptomatic infection. So if we wanted to hit herd immunity threshold with a vaccine with only 50% efficacy, we need everybody vaccinated if we're going to get there. Let's talk about some of the specific vaccines and the immune response. So many of you may have been hearing some of the issues about immunity and whether it lasts or not and whether it fades or not. Most of the vaccines and most of the immune response has been targeting the spike protein. This is the surface molecule that is responsible for attaching to our cells and gaining cell entry. If you look at how many of us will make antibodies and in what span of time, the majority of us will make antibodies somewhere between 10 to 20 plus days. This is IgG response in people who were infected out 0 to 5, 6 to 9, to 10 to 13, 14 to 25 days. Looked at another way here. This is a dot graph of all the subjects. And you can see most of them by about, day 14 to 18 have crossed that threshold of detectability for antibodies. So most of us do make an antibody response. But there are reports starting to circulate that they maybe don't last very well. This was one paper in the New England Journal looking at the half-life of these antibodies being as short as maybe 36 days. Another study that was published in Nature looked at declining antibodies as early as 8 weeks after infection, and this is the acute and convalescent antibody levels in subjects. Put another way, this is the acute phase, here's the convalescent phase on the bottom, and I just point out in asymptomatically infected patients, up to 40% had lost their antibody levels by 8 weeks. You know, this has made some pessimism in how likely we are to be able to mount a good immune response that's protective over time. But I would contrast that. It sort of depends on how you look for antibodies, what assay you do, how you do these, and I would say the jury is not out on this. This is Iceland, that just published a study in the New England Journal of Medicine, and they looked at their subjects out to 4 months after infection, and they found that still 91+% were still antibody positive at 4 months after infection. And this was with what they call a pan-IgG antibody assay, looking at a number of different SARS-CoV-2 antibodies. So I think the jury is still out on the durability of antibodies. I'm fascinated by this study that was published in Cell. This looked at antibodies, which is the blue line, and it looked at T-cell responses, which was the red line, in a control group, which was blood donors from 2019. Here, no one had T-cells, as you would expect. 2020 blood donors, and I believe this was, I'm trying to remember, I think this was up till about like May of this year. And here they found 2020 blood donors. Sweden, remember, is the country that said, let's kind of let it go. They actually didn't really do that, but they let it go more than most countries. 13% of their blood donors it's either late April or early May, were seropositive. But in that same group, 29% had T cells. So when you look for a different aspect of immunity, a lot more. And here, exposed family members. This is interesting because when we think about family members who get infected from someone in the house, we kind of say somewhere maybe between 20 to at the most 40% may develop COVID. Here they found antibodies detectable in 60% of exposed family members, and T cells in 93%. So maybe most family members get some degree of infection if you look hard enough for it. And if you take people who are mildly ill, that gets up to 97% have T cells, and severe illness, all of them have both antibodies and T cells. So maybe when we look a little harder at different aspects of immunity, it may be much better than we think. And in fact, with vaccines now, most of the vaccine candidates we're looking at, this is just one study of this. This is the Moderna vaccine, a messenger RNA vaccine, looking in rhesus macaque monkeys. They make a very nice memory T cell response, which is what we like to see for long-term protection against viruses. So that's promising. So we have sort of some competing evidence. So there's evidence for and against durable immunity. Against durable immunity is that you see no durable immunity in 3 out of the 4 seasonal coronaviruses. So the common cold viruses we're talking about, reinfection is common, durable immunity is not. Select antibodies, as I've shown you, do show waning as early as 3 to 4 months. We've now had several SARS-CoV-2 documented reinfections. And we see that asymptomatic and mild infection mounts a lower immune response, except maybe when you look for those T cells. For durable immunity is that, again, you see durable T cell immunity in other viruses like this, MERS and SARS-CoV-2. There's been recent studies that show people that were infected with those years ago still have good evidence of T cell immunity. And if you do like Iceland did, pan-IgG antibodies, you can find antibodies out to 4 months. And several of the vaccine candidates are shown to induce both B and T cell responses, including memory T cells. vaccine and natural infection, at least in the short term, this isn't maybe, we don't know about durability here, but at least in the short term, protect rhesus macaque monkeys, a very good animal model. And so far, yes, there are documented reinfections, but it still appears to be a rare event. I mean, there's now been tens of millions of infections worldwide, and we can count on one or maybe two hands the number of documented reinfections. That may change as some studies look at this. We don't know yet, but so far that does appear to be rare. So where are we at with vaccines? New York Times has a very nice website called their coronavirus vaccine tracker. I'll kind of focus on phase 3, where there's currently 11 vaccines in trial. There's been limited or conditional approval of 6 vaccines. That's been in Russia and China, with, mainly based on phase 2 data, which we would not do here in the United States. No vaccines have yet been approved for full use. These are the front runners, I think, right now. And the front runners aren't necessarily the ones that are going to finish crossing the finish line. These 3, I'd say, are the front runners for the US: Moderna, Pfizer, and the AstraZeneca Oxford vaccine, which is mainly being driven out of the UK. But there are a number of others that are in phase 3. Another one that's sort of being looked at a lot for the US is this Johnson & Johnson vaccine. And we'll talk about a handful of these here. So when you look at the different vaccine strategies that can be employed for SARS-CoV-2, there's a variety of platforms, new platforms of messenger RNA vaccines and DNA vaccines. There's been no licensed vaccine with one of these platforms yet. Replication of defective viral vectors, I'll talk about, replicating viral vectors, whole-killed viruses, live attenuated viruses, and purified viral protein. These 5 try to take advantage of making our own cells make components of viral antigens, because when our body is forced to do that or engaged to do that, we tend to make a much better immune response. When our cells get involved in the process of either making a defective virus or a part of a virus, we tend to make a better immune response. Whole-killed virus and purified proteins tend to be just antibodies and not as good on the T cells. Let me talk about these 3 here. So messenger RNA and DNA vaccines are a new platform. It's where you actually inject a piece of DNA, like in a plasmid DNA, or a piece of RNA. DNA actually then is just has some genetic material for the spike protein. The idea is that you will get that DNA taken up into the nucleus of the cell. So these are in the dendritic cells of the skin, can be maybe the muscle, and it gets taken up and it makes its own messenger RNA and viral proteins, and then those get released and makes an immune response. To get the cell to take in that DNA, you have to do kind of a funky thing here with a new device that's being kind of looked at called electroporation. You got to kind of electrify the cell a little bit to open it up so that DNA can get in. RNA vaccines take a little piece of messenger RNA that inject, and that RNA gets expressed into viral proteins and makes those for a brief time, or there's some methods now to try and make it do that for a little while. It does not get taken up into the genetic material of the cell. The RNA vaccines that are really the front runners in the US right now, the messenger RNA vaccines, are the Moderna vaccine and the Pfizer vaccine. Moderna has completed their enrollment, so they're done in their phase 3, and now they're in their observation phase. Pfizer is getting very, very close to completing its enrollment. Pfizer released some preliminary data on their enrollment and made the point that they've been able to do a fairly good job of enrolling a lot of the minority groups that have been talked about as really important, because these groups tend to bear a higher burden of disease. So they've got about 10% enrollment of Blacks, 13% Hispanics, about 0.8% of Native Americans. We could probably do better there, 5% Asians, and they've got 47% in people over the age of 56 up to the age of 85. And I think Moderna is looking pretty similar to this in their enrollment of minority groups. So the pros and cons of messenger RNA vaccines: they can be made quickly, they don't cost much, they don't require an adjuvant, They're non-infectious. They're synthesized by in vitro transcription, so not hard to make. They're free of microbial molecules, so they don't really have much in the way of impurities. They don't get taken up into our own cells. They're not integrated into our cells. They don't become part of us. And they induce very good both B and T cell responses. Disadvantage is they're new. We don't have any experience with them yet. There are no approved RNA vaccines out there. Some clinical testing for other viruses like rabies and influenza has been done. They're not very stable. These are the ones that need this deep cold chain to be preserved and then thawed, and you got to give them very quickly. So they make technical challenges for transport and delivery. They can have an inflammatory reaction at the injection site. There can be potential difficulty of intracellular delivery. Development of additional technologies for storage and administration is kind of in the works. Most of these require, as I said, a deep cold chain for longevity and stability. They tend to be not as immunogenic as other types of vaccines. They are likely to require multiple doses. Most of these are being studied with 2 doses. It's possible they may need a booster in the future as well. We don't know yet. I'm not going to spend a lot of time on the DNA vaccines. That, as I said, gets taken up into the nucleus. Inovio is the company that's making that one. They have to also license this funky device that does this electroporation to get the DNA to cross through the cell membrane. It has similar advantages to the RNA vaccines. The disadvantages are also similar. And the fact that it's just a little weird that it's taken up into your own host genome. You know, what does that mean for long term? It should be pretty limited. It's not going to, those cells, you know, shouldn't replicate a whole bunch, but it's a little odd. And it may require an adjuvant and these special tools for delivery. Other platforms are replication defective viral vectors. This is probably thought to be the most likely best ultimate vaccine candidates. They're in phase 2 and moving into phase 3. These use typically adenoviruses that don't cause much harm to us. Sometimes they're a chimpanzee adenovirus that doesn't replicate very far in us, but it will infect a cell. So you take this wild type adenovirus, you take out the genes that allow it to replicate, and you insert a gene for the SARS-CoV-2 spike protein. It infects our cells, starts to, if it's the wild type, it'll start to make other cells, but we don't do that wild type. We use this modified type, and it won't make new viruses, but it will make spike protein. And it won't replicate or spread to other cells. These mount a very good immune response. We have some experience with these. They tend to work well. The AstraZeneca Oxford vaccine is one of these. The Johnson & Johnson vaccine is one of these. There's a Chinese vaccine, and one of the Russian vaccines is this platform. Advantages of these is that they amplify antigen production. You get a lot of antigen made, and so you get a nice immune response, both good B and T cells. I can't get into, limited on time here, but there are some worries about something called antibody-enhanced, antibody-dependent enhancement, where you can get kind of a funky immune response that actually can make infection worse. This has mainly been seen with the dengue vaccine. It's a theoretic problem. It hasn't really been seen, and it was seen with a candidate RSV vaccine way back. If you have a Th1, this is T cell type 1, thymus type T cell response, that almost never happens, and that's a better immune response. And what we're seeing with these is that's what it's biased towards. That's the good thing. It only needs refrigeration. It often may only need 1 dose. There's a fairly good track record with these. Disadvantages: cross-reactive adenovirus antibodies. So we can get infected with adenoviruses, and if we make antibodies to those adenoviruses, they may knock out this vaccine adenovirus before it has a chance to take effect. The antibodies to the adenoviruses may make future booster doses less effective, And these are also typically grown in cell cultures that have been derived from past aborted fetus. There's a handful of cell cultures that are derived from single abortions in the past, and that is sometimes considered to be morally objectionable to some groups and some persons, and that can be an issue raised by some potential consumers of this vaccine. I'll briefly mention replicating viral vectors. This is like one of the current Ebola virus vaccines. And so you take a virus that's pretty harmless to humans. The one here is vesicular stomatitis virus. You take a SNP out of that and insert the viral protein, the gene for the viral protein you want expressed. You then inject that, and that will replicate. And that makes, because it's a replicating virus for at least a little while, it makes a very good immune response, and it expresses that surface protein. This has looked pretty good for Ebola. There is a candidate vaccine from Merck along this lines. I think it's still, it's still in phase 1, so it's pretty early on. Purified proteins is a common thing. That's like the hepatitis B vaccine as an example. One of the platforms for this is the baculovirus-based influenza, like we have with the flu block, the cell culture flu vaccine. You take a baculovirus, and that's tropic for an insect cell culture, and so you can grow it in a vat in these insect cells, but you insert into that an engineered virus that carries the genetic material for the protein you want expressed. Again, this would be the spike protein, and you got a vat churning out spike proteins, and then you can purify that and inject that. Sanofi GlaxoSmithKline collaboration is moving into phase 2 with that. These tend to be very safe vaccines, not quite as immunogenic. They tend to be biased more towards a B cell antibody response than the T cells. In the last few minutes here, I want to talk about what does, warp speed, Operation Warp Speed mean. So I think, the thought from the White House was that would instill confidence in how, great they were, moving in progress. I think it's actually kind of freaked people out a little bit, like you're taking shortcuts and cutting corners. Well, what does it really mean? So if you look at the traditional vaccine development pipeline, we have sort of the exploratory phase where you're doing stuff in the laboratory and in the test tubes until you, for example, identify what you need to get out of cell cultures, purify your product. Then you start testing it out in animals. You want to see that you can get a good immune response in animals, that it seems safe in animals, that it protects the animal from a viral challenge. And if that all looks okay, the company, the pharmaceutical company, files for what is called an investigational new drug application to the FDA. If the FDA says, yeah, we think you're doing a good job of reproducibility in your laboratory processes, you've got pretty good animal data, you can go ahead and start clinical trials on this. If that's the case, it moves into phase 1, where you have somewhere maybe around a couple dozen to 100 subjects, where you look at, did it make an immune response in these healthy subjects? And did we get any major safety issues that came out? If that looks good, then it moves into phase 2, where you typically enroll hundreds of subjects. Here you're looking to find the lowest dose possible that will get you still a good immune response, and you're looking harder for any safety issues. If that looks good and you don't have any major safety signals coming out of that group, you move into phase 3, which is typically thousands of subjects. And here you're looking at your target population, and you're looking harder now for safety problems, and you're looking for actual efficacy of that vaccine compared to either a placebo or whatever is the standard of care. If that all looks good, they apply for a biological license application to the FDA. The FDA looks at typically tens to hundreds of thousands of pages of paperwork for months. And if that all looks okay, they will grant that license, and then the company starts to manufacture it. They have to ramp up their manufacturing and then develop a distribution pipeline. This is typically, just to get to that license, is typically 10 to 15 years with about 90% failure rate somewhere along the way. 50% of that failure rate is in phase 3. So this is a big risk for pharmaceutical companies. Most of these do not make it through. And we have to have a word of caution. We see all these vaccine candidates coming through, 90% of them fail. So we're hopeful, we're optimistic, I'm optimistic. We might be in for this for a long time if these vaccines start to fail in phase 3. That's where we're at for several of them right now. What's Operation Warp Speed? What's this acceleration? Well, it's a few things. It is not curtailing this important part where we get our safety and efficacy data. What it has curtailed is this exploratory and preclinical phase, because most of it was done already. Because of the threat that we knew from SARS, CoV-1, and MERS, lots of research pharmaceutical companies had been working on these platforms and doing the laboratory methodologies and the animal studies that allowed them to very quickly flip this to a SARS-CoV-2 exploration. So that went very fast. Then we've been starting these clinical trials, which there has been some layering of one on top of the other. So they may combine phase 1 and phase 2. So there's overlapping enrollment as and ongoing review, but they aren't cutting any of these phases out. And then instead of the FDA reviewing it at the end, the FDA is doing a rolling review and approval process as they kind of go through each of these phases. And then the federal government has said, we know we can't wait for you to start manufacturing at the end of all this and waiting for approval. We want you to start making it now so that it's ready to go if it proves to be safe and effective. So they've pumped billions of dollars into production of these vaccines, and we're even getting word that we may get them in our states as early as a couple weeks from now, not to give, but to just have ready if it proves to be safe and effective. So this is what's been shortened, this rolling review, the manufacturing taken out of the risk for doing that, taken out of the hands of the pharmaceutical companies and borne by the federal government. And this has condensed all of this typically very long process into maybe 1 to 2 years. I mean, we've been at this, since February, March. We may have a vaccine by the end of this year, early next year, maybe. These may fail, and we may be waiting another year for one of these to make its way through. But that's a greatly condensed timeline without really sacrificing the safety and efficacy data that we need from the typical FDA process. Now, you may have heard a couple of these vaccines were halted in clinical trials. The AstraZeneca Oxford vaccine was halted because of a case of transverse myelitis, that's autoimmune spinal cord inflammation in the UK, and more recently a death in Brazil in the trial. The US halted, there was a pause everywhere, and then it was restarted most everywhere. It was still halted in the US and just restarted, I think, last week sometime. The death in Brazil, most everybody thinks was in the placebo group because that didn't get anything stopped. And if it would have been a death in the vaccine arm, they usually will pause that for a more extensive review. They've done apparently a deep dive on the transverse myelitis and have felt okay to proceed on. Johnson & Johnson had some serious adverse event. They haven't released what that was. That trial was put on pause. This is common because well, I'll get to this in a second. That trial has restarted in the US as well. But is that overly worrisome? Well, if you think about the size of the populations that are being enrolled in these trials, it's about the size of like Minot, which is about 40-some thousand, I think. That's what they're aiming to enroll in a lot of these. Well, what might you expect over a year or at least half a year or whatever in a city the size of Minot? Well, for transverse myelitis, you might expect about 1 to almost 5 per million. That would be maybe 1 case of transverse myelitis every 5 years in a city like Minot. So having a case in this group is a little bit worrisome. But if you include multiple sclerosis, which can cause transverse myelitis, that's about 25 per million. That would be about a case every year in a city like Minot. So that's just background. So we shouldn't get overly anxious about a report like that until we see it's clearly higher than the background incidence, like we talked about a bit ago. So the other thing about this is that we're likely to have an emergency use authorization rather than the standard FDA process. Emergency use authorization happens when there is a federal emergency declared, which we have, and that can expedite the process through the FDA, and that authorization will last as long as that emergency is in place. That process is reviewed, and the FDA is advised by a group called the Vaccine and Related Biological Product Advisory Committee. This is a powerhouse of independent experts on vaccine safety, clinical trials, coronavirus epidemiology, virology, etc. People on this committee are people like Paul Offit, who is one of the world's authorities on vaccine, a very staunch advocate for vaccine safety. Another person is Dr. Archana Chatterjee. She used to be at Sanford and Sioux Falls, now is at the University of Chicago, a good expert in pediatric immunizations and in clinical trials. These people are very vigilant and concerned about making sure we have a safe vaccine that is delivered and that these processes are not shortcutted. That group will review and advise the FDA. This EUA is faster to obtain, and the FDA can authorize this as an unlicensed product, compared to a typical biologic license application, which can be up to a year just to review the data. It's it's a larger multidisciplinary FDA review team. They inspect the vaccine's manufacturing plants, and it involves the VRBAC. But this is a streamlined process here. But this group, which is very, very good at this sort of thing, will be heavily involved. And the FDA has promised that they will at least assure 2 months of observational follow-up after these phase 3 trials. There's actually still some back and forth whether that should be longer. They set that 2-month follow-up because we know from experience about 90% of serious adverse events are detected in the first 6 weeks after vaccination. So we have to weigh the safety of the vaccine against what we know is happening with the pandemic to us now. I think these are reasonable measures. There are some people that want a higher safety bar than this. I think this is, in my personal opinion, this is reasonable. Now, last, I just want to finish with what North Dakota is doing. Some of you have kind of gotten the mistaken notion that we are getting vaccine first. No, I kind of wish that were true, but no. We were picked to be 1 out of 4 states to pilot a distribution plan. What might you do, and how would you do it, and how would you model it if you were to get vaccine by, say, November? So Molly Howell has been working very diligently and very hard with a large group of stakeholders around the state to develop a distribution plan for the vaccine. This is not a vaccine clinical trial. It is not having any group or population or segment of our state as guinea pigs for a vaccine. It's how will we give it out once we get it. We were chosen as one of these states because of our rural nature and our tribal relationships, which were thought to be important for any ultimate plan. The North Dakota goals in this pilot were: how would we ship vaccine out to North Dakota? How would we safely and effectively transport, store, and distribute the vaccine? What about the temperature issues, thermostability? How about administration, especially if we're using things like multi-dose vials where we might have to unfreeze something and give the vaccine very quickly to large groups of people? How would we track the distribution, and who would administer the vaccine? It's following roughly the Advisory Committee on Immunization Practices from the CDC's tiering, which is going to, in the first groups, involve healthcare personnel in Tier 1 and Tier 2 groups, other essential workers in Tier 1 and Tier 2, people with underlying medical conditions and people over the age of 65, vulnerable people, and there's various tiering of each of these groups that I don't really have time to get into. Other ways of looking at this from the ACIP has been to recognize that there's maybe overlap of some of these groups: healthcare providers, essential workers, and those who have in those groups or high-risk medical conditions or the elderly. And later groups being racial and ethnic groups that may be at higher risk. And ultimately, we're kind of looking at essential workers, healthcare workers who might especially have some of these higher risk groups, because we want them to be able to be safe and working and still on the front line. An interesting additional safety measure that is being developed is called the Vaccine Safety Assessment for Essential Workers, VSAFE. This is a text messaging or email from the CDC with follow-up daily for the first week post-vaccination and weekly out 6 weeks. Any clinically important event will be reported by the vaccinated person, and follow-up from the CDC on clinically important events will be completed through the VAERS, already existing known reporting system. So it's an extra layer of monitoring and vigilance. Our estimates here in North Dakota are that we've got about 550,000 people with one or more of these various risk factors. We've got about 34,000 essential workers, about 68,000 healthcare workers. We're looking at phase 1A, healthcare workers, long-term care providers and residents, direct care public health workers. Then moving into phase 1B would be tribal groups, high priority infrastructure, ancillary healthcare workers, high risk medical conditions, and adults over the age of 65. All right, I will stop there. I don't know if it's Molly or Lauren, you want to talk about the CME post-training assessment for getting your continuing education credits?
Absolutely. I can talk about this. So we are offering 1 credit of CME or CPE for attending this session. This is the information for the CME assessment. If you are applying for this, you need to follow the information stated in this slide, and I will post the link again in our chat box. Dr. Carson, do you want to go on to the next slide?
Yes. Yeah, go ahead, Lauren.
Oh, yeah. This is, I guess, this is just the same. Dr. Carson, do you want to go to the next one?
Maybe I didn't.
Sorry.
Oh, yeah, we updated. Sorry about that. Oops.
Okay, so this is the information for CPE. That's for pharmacists applying for 1 credit of CE. This has to be completed by November 28th, but I wanted to note that we will be posting these sessions on our center's webpage, and it will be continuous education for around a month or 2. So if you are unable to apply for credit now, we will be giving that option on our website.
All right, one last poll.
Yes, I will. Let's see. If the FDA authorized a vaccine to prevent COVID-19 today at no cost under EUA, would you agree to be vaccinated?
Great. Well, it looks like a little bit of a bump up there in the people who said yes, and I think maybe a little bit of a decrease in those who said no, and still a chunk that are unsure, which I think is reasonable and understandable. And I think I personally I'd like to see the actual data that the FDA gets to review, but I do trust the process, and I trust the people that are doing that, as long as we don't see some outside intervention in that FDA process. And so far, I think it's looking good that way. So I'll stop there. I'm sorry, we went, oh boy, we went a fair amount over. Sorry about that. But if the people want to still hang on, or if they're able to and don't have to jump back to work, I'd be happy to try and work our way through some questions.
Yes, feel free to post your questions in the chat box.
Person, one of the questions we had was related to any COVID vaccines in kids.
Yeah, so I see that Pfizer is talking about extending their vaccines down to the age of 12. And I don't know when they're going to start that, but it sounded like that might be relatively soon. There has been a number of discussions that we really ultimately can't think very well about the idea of herd immunity without involving children. There are a couple other vaccine candidates, not ones that are being trialed in the United States, but in other countries there are at least 2 other of the vaccine candidates that are being trialed in children. There's debate about whether you can extrapolate adult data down to children. I think most people are a bit uncomfortable with that idea. But I think if we can get the high-risk groups vaccinated first, my hope would be it would expedite its way through pediatric trials fairly soon after that.
I can see we have this question in the chat and we got it in our email. Have you seen any genetic shift or drift observed in coronavirus that may prompt, or similar to like a influenza vaccine and vaccination?
Right. So yeah, the question being, does SARS-CoV-2 mutate in a way like similar to influenza that shifts so much that we need to change the vaccine up almost every year? The answer to that is no. It does not mutate at the same pace. It has more replication integrity than certainly influenza. There has been some shift or change, and there's this what's called the DG mutant variant. That does not seem to be affecting immunity or immune response to the spike protein. It seems to be just as robust. So right now, I think most of the experts feel that current vaccine candidates with a little bit of minor variation and mutation that they've seen in the virus is not going to be a problem. Now, could that progress further over time? Yes, possibly, but so far it looks pretty good for a durable immune response that may not be hopefully subject to the same kind of vagaries that happen with influenza.
Have you heard anything about challenge trials, Dr. Carson?
Yeah, I think that the UK is talking about this, and I actually think they're planning to do this. So challenge trials are an ethically interesting proposal, and this is where you take a healthy small group and actually let me back this up. So to see efficacy in the vaccine with a vaccine in the way we do clinical trials typically means waiting till you have enough people get sick in the trial to see if you've got a significant difference in acquisition of infection between vaccinated and non-vaccinated. Well, Pfizer's been at it a while here, and they've just reported, that they're waiting for their first 32 infections out of their 30-some thousand subjects, and they haven't hit it yet. And that's to hope that you might have like 26 in the placebo arm and 6 in the vaccine arm. That would be a 75% or so vaccine efficacy. And we're waiting. You don't want infections to happen, but you kind of do in those trials. Well, you can circumvent that all by doing, just like you do in monkeys, you can do a direct challenge in humans. You can say, hey, any volunteers who will sign up to get the vaccine, and then be directly challenged with the lowest inoculum of virus that we know can establish an infection. And that will give you efficacy data, direct, overt efficacy data. There are ethical issues with that because this is a virus that can make people very sick. Not too often in young and healthy, but it can. But as I said, the UK is planning that from what I hear, and there's been other people that are talking about that. Even some very prominent ethicists like Arthur Kaplan in the United States has advocated for those kind of trials in a situation like a pandemic like we are in. Other questions?
Another question.
Sorry, I got a kid back here. Should those who have already had COVID get vaccinated?
That's a great question. I do not know. I think we're going to have more information about the immune response of like the asymptomatic to minimally symptomatic persons as we explore like T-cell responses more. We're going to be doing a big study on this in North Dakota over the next year. I think we need to see a lot more about the reinfection data. So on the one hand, it appears to be pretty rare, but to prove reinfection is kind of a hard thing to do. So there might be a lot more reinfections out there than we know about. We are hearing, for example, in our nursing home staff of people testing repeat positives after 3 or 4 months after they had it earlier without any symptoms. Is that a real reinfection? We don't know. So I think if we learn that that happens more often than maybe appears right now, it'll be recommended to get the vaccine. I would say the likelihood of people being recommended to get the vaccine, even if you've had it, is probably pretty high, but that's yet to be determined. Maybe Molly knows offhand. I don't think people are being excluded from the vaccine trials if they've had the infection already. Do you know, Molly or Kylie?
Not that I have seen, no.
If they've had a handful of subjects that had it before, I don't think that's an exclusion criteria. That might give us a little information.
I believe yesterday during the Pfizer trial, I think I heard, they said it wouldn't exclude. They asked if I had had COVID before, and they said that would not necessarily exclude you.
Other questions? I'm seeing lots in the chat.
We have another, yep. So is there any information yet regarding who will be eligible to give the vaccine? For example, will it be supplied to community pharmacies for pharmacists to administer?
I think the short answer to that is yes, but Molly, I'm going to let you handle that question because I think you've done a ton of planning around this.
Yeah, so any provider who's authorized to vaccinate can enroll with the state of North Dakota to receive and administer COVID-19 vaccine. Now, it's likely early on vaccine will be extremely limited, and so we will prioritize providers who vaccinate those populations. For example, if it's healthcare workers or long-term care workers, we would prioritize providers who vaccinate those individuals. And also, some of the vaccines, as Dr. Carson mentioned, have very specific storage and handling requirements. And so there may be some limitations based on those storage and handling requirements as well as to who can receive vaccine. But any provider who is interested in potentially vaccinating for COVID-19 vaccine can go to our website and complete the enrollment process. I do want to mention that last week and then also earlier on this year, HHS has authorized pharmacists, pharmacy interns, and pharmacy techs to vaccinate. And so those are pharmacy techs, especially for North Dakota. That's a new type of provider that after certain training for immunizations and being CPR certified will be able to administer COVID vaccine.
I see a question that was in the chat box about a study that showed T-cell immunity to COVID-19 in, from 10-year blood donors from 10 years ago. Is there cross-reactive T-cells from prior coronavirus infections? I need to look at that a little more carefully, and I've seen some chatter about that as well. In the study that I showed in Cell, from the journal Cell, those 2019 blood donors had no T-cell response, specific T-cell response in the way they did the assay. There's different ways of doing this assay. They showed 0 had T-cell immunity to SARS-CoV-2 in the 2019 blood donors. So I'm doubtful that prior exposure to the circulating coronaviruses gives us any, and there's been other studies that suggested it really does not look like it gives us any level of protection against SARS-CoV-2. Other questions you want me to field, Kylie?
Yes, I guess we had another one that we had received where it said, will the efficacy of each vaccine be provided before people receive it, or will it be known?
Boy, I sure expect so. Yeah, I mean, absolutely. The FDA has said they'll be transparent about this data. They even said that they were going to release interim, the data safety monitoring boards that will break the blind when you get X number of events. So like when you hit 32 infections, they break the blind and analyze, do we have enough difference between the two groups? And when we get to 64 and when we get to, and so on, they've said that they were going to make that even transparent, which makes me think, we haven't heard that on any of them yet, which makes me think they're not getting enough cases yet to be able to break the blind. But we have been told we will get that efficacy. I sure want to see that. I absolutely want to see that. So I would expect that. And in the VRBAC, committee meetings, they've been all about like we need to be very transparent. We need to be very open about where we're at with safety and efficacy on this, because they know trust in vaccines is on the line with this vaccine. Like trust in vaccines for like all future vaccines is on the line with this process.
Another question we received in our chat box: do we have clear information on if a person is given the first dose but develops COVID while they are waiting for their second dose? Assume they should still be given the second dose. Just curious if there has been discussion or concerns with that possibility.
I don't know the answer to that. I presume that they would continue in the trial. Molly or Kylie or Lauren, do you know the answer to that?
No, but I'm sure that's something that the Advisory Committee on Immunization Practices, when they make their recommendations, will take into consideration.
Yeah, I mean, that's probably going to be a small subset, but because I think the interval is 28 days for the 2 lead candidate vaccines, if I remember right. So, there's maybe a handful of people, but it should be pretty small that, happen to land their infection right in between the two.
We also got a question through our CIRE email, and is what do immune profiles for immunized versus wild type infected individuals look like? How difficult or possible will it be to determine if someone was infected versus immunized based on sero tests?
Yeah, my understanding is that the immune response to the vaccine looks as good or better than the wild type infection, especially if it was asymptomatic or mildly symptomatic. The immune responses in some of those asymptomatics hasn't been really high, especially the antibody levels. So the vaccine responses have typically been better. The RNA vaccines, they're not quite as immunogenic as, like I said, the non-replicating or replicating viral vector vaccines. So they're comparable to, I think, slightly better. But if you're asking, could we tell, down the road after vaccine starts to get distributed, could we tell if somebody's got an immune response that looks like it was from natural infection versus vaccine? I don't know that would be possible.
We have one more in our chat box. Do you happen to know if the shelf life of the vaccine is about at the 14-day mark after opening the vial? Also, do you happen to know if all of the vaccines are to be required to be stored in a negative 80-ish area?
Yeah, so I'm gonna let Molly answer. She knows the shelf life question a lot better than me on all these. No, not all of them need this minus 80 storage. In fact, I think it's Molly, you correct me. I think that might be the Pfizer vaccine. The Moderna, I think, is minus 20. And the other ones, a lot of them, there's going to be some of the, like the pure protein derivatives, probably going to be lyophilized product that you can reconstitute. The viral vector ones are refrigerated cold chain. So Molly, do you want to comment on that further?
Yeah, sure. So yes, the Pfizer vaccine is the one that requires ultra cold chain storage down to potentially negative 80. That vaccine has a stability in the refrigerator at 120 hours. And then the Moderna vaccine, which is one of the another mRNA vaccine, that requires frozen storage, so normal freezing temperatures. And that's the one that has the 14-day time period in the refrigerator. So if you put the Moderna vaccine in the refrigerator, then you have to use it within 14 days.
Thank you. I see there was a question in the chat box about vaccination for pregnant women. I'm pretty sure right now pregnant women are excluded from, I think, all of these trials. Do you know any different on that, Molly?
That's correct. That's my understanding as well.
Yeah, so that's probably going to be a contraindication to vaccination, at least for the short term.
We also had a question about, let's see here, please comment on progress made with the development of AC2-based pharmacological therapies.
Yikes, I don't know that. So the ACE2 receptor is one of the co-receptors to which SARS-CoV-2 spike protein attaches, and there's there's been, talk about targeting that interaction. And I should know this, you caught me here. I should know if there's any candidate drugs in the pipeline that are specifically aimed at that interaction. The Regeneron, the monoclonal antibodies that are being developed, that is targeted at blocking that, interaction. But frankly, I don't know offhand. I'll try and look that up, and maybe we can answer that on the CIRI website. And I know Dr. Nagpal just gave a talk on therapeutics. I don't know if he looked into what was in the pipeline there, but and if somebody else knows, please chime in. I'm happy to have somebody else help answer that.
We also got a few more questions in the chat box. So would you assume breastfeeding persons would be excluded as well?
I would assume yes. I haven't looked, boy, I should have looked a little harder at the exclusion criteria on the vaccine trials, but that typically is the case in these, first round of trials. They may sometimes, like with pregnancy, sometimes people find out they're pregnant after they were vaccinated, and they'll often do a registry, and follow those. Breastfeeding is another matter. I mean, if you're breastfeeding or not. Again, I'll have to ask Molly if if that was an exclusion. Molly, my bet is yes.
But I do not know the answer to that. No.
Dr. Carson, it is
an exclusion.
Yep.
Okay. Thank you.
For Pfizer, anyway.
Okay.
Another question from our chat box. This is not about the vaccine, but the PCR testing. We had an employee recently test positive for COVID. She tested positive 5 months ago. She has been asymptomatic both times. Creates a hardship when off for 10 days each time and additional for children. What are your thoughts?
Yeah, this is really tough, and I'm hearing like story after story after story of this. I'm a medical director of one nursing homes, and I think we've got we're in that one nursing home, we've got like 8 similar stories. So you have to imply one of a couple things. You have to, I'd say one of sort of 3 things. Either it's the same virus and scraps of RNA still hanging around and was missed on one of those middle tests. So those middle tests were sort of a false negative, and now you're picking it up again. Or it's a new infection. Or one or the other of those tests was a false positive. The problem is you can't assume, we just can't assume, especially in a long-term care worker, that it's a false positive or that it's the same infection and that they're not infectious anymore. We need better data on this. So here's what we really need is we need those people and those scenarios to submit samples for viral culture to see if it's culturable virus, which is a surrogate for transmissibility. I hope some research group is starting to try and attempt this out there. But in the interim of having better data on that, we cannot assume that they're non-infectious and put potential very vulnerable long-term care residents at risk. I know that's frustrating. I know that's a hardship, but I think we're stuck.
Okay, a couple more questions. Do we know if there's a minimum age to receive the vaccine? I haven't heard of any clinical trials taking children. So if they aren't tested, how will we know it's safe to give it to them?
You don't. So, I mean, they typically for our FDA approval of vaccines, it has to be tested in the age group before you can give it. We don't give really off-label vaccines to different age groups. So it really would need a pediatric trial. As I said, the Pfizer vaccine is looking at extending their trial group down to age 12. And I saw 2 other vaccines not in the US are looking at down to age 3. So, and ultimately, I do think we need to involve children if we're going to look at reaching herd immunity. We can't have that big of a segment of the population as potential carriers and spreaders and achieve that. But I think that's going to be down the road. So realistically, for us to kind of look at herd immunity through vaccination, I mean, that's a year or 2 or 3 years away. Maybe a combination of vaccinating the vulnerable and prior natural infection might get us closer. But I think we do have to be a bit realistic here that we may be a ways away from that. But hopefully we can get individual level protection for the most vulnerable and cocooning the most vulnerable with the workers that are around them.
We have one final question. Is there any other data? Let's see here. Sorry, it popped up. Few more questions popped up. Is there any other data about using a vaccine with other vaccines, such as influenza?
No, and I think the candidate vaccine, COVID vaccines, have required that they not receive influenza with. Molly, you'll know this better than me too. Is it like they don't want them to have it within 30 days prior or after? Do you know offhand?
I'm not sure about the specific time period, but we have been told that COVID vaccines will have to be administered independently from other vaccines, but I have not seen the specific time period.
As a general rule of thumb, there's not much problems with giving vaccines together at the same time. Usually that's not an issue, except in the case of 2 live virus vaccines. Now, these are a new platform. That's probably why we're probably going to be pretty careful about that. You know, like the RNA vaccines, it's a new platform. So, you know, I'm sure they want to be extra cautious about anything that might interfere with mounting an immune response. So I think that they're going to have an indication that is not to be given with other vaccines in close proximity. I have to jump to another meeting. Maybe one last question. I'm supposed to be on another meeting with our university here at 1:30, which it is right now. But is there a last question, Lauren, I can try and knock out? And maybe Molly can stay on and Lauren and Kylie, you know a lot of this stuff.
Sure. If you would like to answer one more question, what happens if the U.S. gets a vaccine 50% effective and a different one in Europe is more like 75% effective? Are we in the U.S. stuck with the less effective vaccine?
That's a great question. I have wondered about that myself. I don't know the answer to that. I think, you know, so like the AstraZeneca Oxford vaccine, the U.S. helped fund that, so we should have access to that one, but it's being mainly trialed in other countries. You know, we've kind of told the rest of the world our vaccines aren't going to be available to you until after us, so you know, we might expect turnabout as fair play here. I mean, China's got several in the pipeline. I don't know how quick they're going to send us any vaccine. The other piece of that question, though, is that I think if a more efficacious vaccine comes along, I think people who got the less efficacious one will be candidates for that vaccine. So I think, as I said at the beginning of this, what we start with might not be what we finish with, but we might start with something, we might accept something in the interest of expediency and trying to stop this mushrooming epidemic right now, but we may land on something that's better down the road that we get revaccinated with. So I'm going to jump off there. I hope maybe Molly and Lauren and Kylie can stay on for another minute or two and answer any other questions. There's still a lot of people. Feel free to jump off. We've gone way over, but it was really good to be able to do this session with everyone, and I hope it was helpful. Thank you.
Thank you, Dr. Carson.
Thanks, Dr. Carson. Okay, Molly and Kylie, you and I will do the last few questions. So, okay, how will the state hospital decide which vaccine to use if many are approved? Molly, do you want to take that one?
Yeah, and so it is possible we'll have more than one type of COVID vaccine available to us. And so some of that will be based on, for example, right now the minimum shipment size for the Pfizer vaccine is 1,000 doses. And so some of it may be dependent on where we're sending the vaccine and who's being vaccinated and if they're able to use the full 1,000 doses. And so we may have to prioritize, for instance, if both Pfizer and Moderna vaccines are available, the Moderna vaccine can be shipped in 100 dose increments. And so the Moderna vaccine might have to be prioritized for more rural areas. Some of it will also be dependent on the providers who enroll, what their storage and handling capabilities are for vaccines. If providers, you know, the ultra cold chain vaccine will probably, if there are more options available, that one will be prioritized for providers who have ultra cold chain capabilities. The Moderna vaccine can be stored in the freezer, so we would try and get that one to places that can accept frozen vaccine. Now, having said all of that, both of the vaccines do have limited refrigerator stability, and so it is likely that we'll have to rely on that refrigerator stability, and we're going to really try, because we know the schedules are different and may be complicated, to give providers only one type of vaccine if at all possible. So we're limiting confusion, but that may not be possible and will be dependent on what we're allocated as a state.
Okay, well, maybe answer a couple more questions. How does that sound, Molly?
I'll
Okay.
try. I'm not Dr. Carson, but I'll try.
Yep, okay, so let's see. The next one is, do you see vaccination replacing quarantine as an intervention measure for exposed, susceptible individuals?
I think we're hopeful that that could potentially be the case in the future. I don't think we know yet, especially because we don't even know what the efficacy of the vaccine that we may get in the future is. And so I think it's really going to be dependent probably on the efficacy of the vaccine. And then it's also probably going to partly be dependent on herd immunity as well, and how much disease is still in the community. And so I think that is yet to be determined. But hopefully, I mean, I think that's what we're all hoping for as an outcome.
Well, I think we've covered all the questions in the chat box. So I think if anyone has any final questions, otherwise we're probably going to wrap it up and call the meeting to a close. And
Lauren, if I can just add, we will post this to the Department of Health COVID vaccine website as soon as we can. And so hopefully by tomorrow, if not, then next week. And so you can go there and we'll send the link out as well if you want to review the presentation. And then I do want to say that this isn't it for education on COVID-19 vaccine, as we do receive additional information about COVID-19 vaccine. And then especially once one is approved for use, we will have additional education for providers in the future.
Great. And I was also going to mention that we will again be posting this on our centers and the NDSU Centers website, and we will be offering the credit for over a month, like next month or two. So if you haven't been able to watch the whole session, you will be able to still apply and get your continual education credits through our on-demand training. So I think that's everything, and thank you guys so much for attending our session and holding out the extra half hour for our questions, but we hope you learned a lot about COVID-19 and the potential vaccines that could be offered our state. So thank you all, and if you have any questions, feel free to email us at the center, or you can, again, just contact us through our email that we've put in the chat box.